Sir, I'm Batham. I'm preparing for NEET 2024 and I'm going to explain the Griffith experiment for the search of a genetic material today. So Griffith started with taking the bacterium for pneumonia, which is Diplococcus pneumoniae, and he took two strains for it. So he took the R2 strain and the S3 strain. Now in the R2 strain, it's a non-virulent strain. Here it does not have a mucus coating over it, while the S3 strain has a mucus coating which makes it virulent. Virulent meaning it will cause pneumonia, while R2 will not cause pneumonia. So Griffith took S strain first, which is the virulent strain. He injected a live mice with this strain, and he observed that the mice gets infected by it and it eventually dies. However, with the R strain, since it's non-virulent, once injected, the mice continues to live. He then took the S strain and he killed it using heat, and due to the heat, the S strain loses its property, which is why once it's injected into the mice still continues living. Now, based on these experiments, he performed his fourth experiment, which he used the living R strain and the heat-killed S strain. Now, according to all of these experiments, he believed that the mice would still live. However, his observations were a little different. He observed that the mice died. And now, on performing an autopsy on this mice, he found that there were a few S strain living bacteria inside the mice. After careful observations, he further concluded that there is a certain principle inside this S strain heat-killed bacteria which transforms this R living bacteria into the S bacteria, which causes the mice to ultimately die. Now, after finding this out, various other scientists performed experiments, and they eventually concluded that it's DNA that gets from this S strain heat-killed bacteria into the R strain bacteria, which eventually forms a polysaccharide coating over it and becomes virulent again.
Very good.